today's lab was about sexual reproduction. And first off, we looked at asexual reproduction, which is reproduction without sperm or eggs. And we just briefly looked at asexual reproduction in yeast, which is a fungi. And yeast reproduces by budding. So you can see I have a budding yeast cell up here. When yeast buds, what happens is you just have a smaller yeast cell grow off a parent cell, and the two cells are genetically identical to each other. So it's asexual reproduction in yeast. It's budding. Reproduction without eggs, without sperm. However, most of the lab we spent talking about meiosis. And meiosis is cell division in order to create sex cells like eggs or sperm. Remember we talked about mitosis last week? In mitosis, you have cells divide in order to create ordinary body cells. And those body cells are genetically identical to the parent cell. Meiosis is different. In meiosis, the cell divides twice to create 4 daughter cells, and those 4 daughter cells are genetically different from the parent cell, and they're genetically different from each other. To understand why, let's think a little bit about human beings. Human beings normally have 46 chromosomes, and if our eggs and our sperm that we made had 46 chromosomes each, when you combined, we'd have a baby with 92 chromosomes, which would be a very, very genetically messed up baby. So the entire point of meiosis is to create eggs and sperm that have half the chromosome number of the parent cell. So if the cell has half the chromosome number, it's called haploid. Has the full chromosome number, it's called diploid. Okay, so the eggs and sperm are created are haploid. The entire purpose of meiosis is to reduce that chromosome number by half to a haploid number. For us, our eggs and sperm have 23 chromosomes rather than 46. Okay, so let's see. First off, let's look a little bit about meiosis and grasshopper testes. So this is sperm making a grasshopper, and I'll pull up a picture of grasshopper testes on the video monitor. There we go. Okay, this is previous slide. These grasshopper cells are in prophase 1. In prophase 1, the double-strand chromosomes, that is the dyads, will line up, forming tetrads, and the tetrads will undergo crossing over, so they'll swap arms of the chromosomes. So you can see you have lots of tetrads forming here. These tetrads are undergoing crossing over. Now, I am not going to go over with you all the details of meiosis. There's another video for that. Also, you should study in your lab manual for that. I'm just going to show you this picture of it. So, crossing over in prophase 1 of grasshopper testes. So, most of what we looked at today was meiosis in the scarus, that is, parasitic roundworms. So, I'm going to pull up a parasitic roundworm slide for you. We're going to try to find some sperm. Okay, so what we're looking at is a pregnant female worm, and she's been cut in half, and she's been fertilized with sperm from a male. So, good. See, these are eggs, and these are sperm. For example, you can see right here there's a sperm that's entered into the egg. So, all these eggs have just been fertilized. But interestingly enough, meiosis has not yet been completed, and the worm is going to continue on for a little bit while longer. Now, our second slide we looked at was scarus bivalent maturation. If you remember in meiosis, the entire purpose of meiosis is to reduce the chromosome number by half. And so in order to do that, when there is cell division, and half of the chromosomes in an egg get kicked off and kicked out of the cell in a small cell called the polar body. The only purpose of that polar body is to reduce the chromosome number by half. Let's find some polar bodies in the scarus. Alright, so here is the female worm. And you can see she's full of eggs. We'll zoom into high power. Okay. So let's find one that's got a good example of polar bodies. Okay, here's a perfect example. This female There's an egg. You can see the sperm nucleus. So there's already been the sperm in the egg. You can see the first polar body is right here. It's on the outer rim of the shell. Here's the second polar body. So the scarus produces two polar bodies. The purpose of those two polar bodies is to remove excess chromosomes. So the egg cell can go from being diploid to being haploid. Okay, let's look at one last slide with the scarus. Or rather, another slide with the scarus. Okay, so after meiosis is completed in the female roundworm, and fertilization occurs, the egg will actually temporarily have two nuclei. These two nuclei are called the pronuclei. One of the nuclei came from the male sperm, the other one came from the female egg. So the cell, for example, has one pronuclei here and one here. One from perhaps the sperm, one other one came from the egg. We can't really tell the difference between the two. So our last slide is scarus cleavage. Now, what's going to happen next is these two pronuclei are going to fuse together. After they fuse together, it's going to create that new single-cell baby worm. And baby worm is going to start dividing by mitosis. And first, that one cell is going to divide into two, and then into four, and so on. So let's take a look at cleavage and scarus. Okay. Some of these cells are one cell, so the pronuclei have just joined and created a new cell. This is a two-cell stage in the scarus. One roundworm cell is divided into two. Let's try to find a four-cell stage. Looks like we've got an example of a four-cell stage here. That's not a particularly good-looking example. So I want you to make sure that you can recognize all the different parts of the scarus meiosis, know what's going on in each of the slides, if you see a photograph of them on the final exam. And the next thing you looked at was starfish development. I've drawn it out on the board, so instead of looking at the slide, I'm just going to come look at it on the board where it's labeled. Come over with me. You should make sure that you can recognize all the stages of starfish development and know their parts. Okay. The earliest stage of starfish development is the zygote. See, the zygote is composed of one large cell. It will divide by mitosis into the two-cell stage, and divide by mitosis into the four-cell stage, into the eight-cell stage. It will divide into a solid ball of cells called the morula. And then it will divide into a hollow ball of cells called the blastula. And finally, it will divide until you have many, many cells. It forms a gastrula. Now, the gastrula is the first time you can see the three cell layers. You have the ectoderm here, the endoderm here, and the mesoderm is right at the end of the cavity. The opening to the cavity is called the blastopore. The cavity itself is called the archenteron. So we've got the three cell layers in the gastrula, the ectoderm, endoderm, mesoderm. I'd like you to be able to recognize all three of those in the gastrula. Okay, so make sure you can recognize all stages of starfish development. The last thing I wanted you to learn was different parts of human female reproductive anatomy. And to start off, this is a fairly late-term fetus model. And you can see it has an umbilical cord with arteries and veins present attached to the belly button. And the umbilical cord attaches to a structure called the placenta, which is itself attached to the uterus wall. So what the placenta does is it takes waste from the fetus's bloodstream, filters them out to the mother's bloodstream, and it filters food and oxygen from the mother's bloodstream to the fetus's bloodstream. So it provides a source of food and oxygen for the baby, and it removes waste from the fetus's bloodstream. The entire structure of the baby or fetus sits in is called the uterus, And attached to the uterus, you have two organs. These are called the ovaries. Meiosis begins in the ovaries in human eggs. And interesting enough, in human beings, meiosis is not actually completed unless fertilization occurs of the egg by a sperm. And if fertilization occurs, that will occur inside the oviducts, these two tubes attached to the ovaries and the uterus. And it'll take approximately a week for that fertilized egg to travel down at the uterus and implant and start developing until it's a fetus ready to be born. So, for your final exam, I want you to study all the phases of meiosis in your lab manual, and also learn to be able to recognize all the stages of the scarce meiosis and starfish development and female human reproductive anatomy. Okay, so now I'm going to briefly talk about sperm anatomy. And so this is what a sperm looks like, roughly speaking. It's got a flagellum or tail. It's got a midpiece, and the midpiece is joined to the head sperm by the neck. And it's got a nucleus in the head. Of course, the nucleus is haploid. It's got half the chromosome complement of a diploid cell. So for a human being, you have 23 chromosomes. At the very tip of the head, we have our acrosome. And what the acrosome does is it contains digestive enzymes. So digestive enzymes help the sperm burrow its way through the membrane surrounding the egg. So it helps the sperm fertilize the egg.